Well, good morning, everybody. Welcome to this week's edition of Micro Seminar. It's my pleasure today to introduce to you Dr. Vikas Yadav. He's a postdoctoral fellow at Duke University with Dr. Joe Heitman. He's been there since 2018. Prior to that, he did a master's degree and a PhD in molecular genetics at JNC Bangalore with Dr. Kaustav Sanyal. And before that, and he did an undergraduate work at northern India at Punjab University. And so, without any further ado, I'm going to turn it over to Dr. Yadav to regale us with tales of fungal biology, an excellent underrepresented set of research and endeavors in micro seminar. So thank you for being here, and thank you for filling in on the fungal side.
Thank you, first of all, for this opportunity, and let me share my screen. So, okay, I think Cameron, you need to switch on my screen sharing. Sorry.
Sorry about that. You should be able to go now.
Yeah. All right. So, good morning and good evening, depending on where you are, to everyone. So today I'll talk about our research work, which basically involves exploring the fungal kingdom for some new and interesting research part. So the title of my talk is Sexual Parasitism in Fungi, and then the follow-up part of it. So I'll start with giving you some basics and then get into what exactly we have done over the course of past 2 years. Now, first of all, the introduction to the model hero of this entire seminar, which is going to be a fungal pathogen. So as Cameron mentioned, my talk is going to be about the lesser focused area of fungal kingdom, which is which lies somewhere here in the tree of life. Now, fungal kingdom basically exhibits different types of shapes, sizes, and there are organisms which are microscopic all the way to very large mushrooms that many of you probably eat. Now, embedded amongst this entire plethora of shapes and sizes, if you go through the fungal kingdom, there is a very small budding yeast that lies, which is known as Cryptococcus neoformans. Now, Cryptococcus neoformans is a haploid yeast which has 14 chromosomes, and it belongs to the file of Basidiomycota. Now, this fungal pathogen is basically present everywhere in the environment. So if you look at the life cycle, you find this in other animals apart from humans, environmental sources like plants and pigeon guano. At plants and pigeon guano, it is actually able to undergo mating in nature, where it forms spores, which are the infectious propagules for humans, and they get into our body via inhalation and starts by infecting lungs, but then can disseminate into other parts of the body. The lethal infection from this fungus is cryptococcal meningitis, which basically infects the layers of your brain. Now, if you look at the disease impact of this fungal pathogen, it basically is present throughout the world at different rates, and the most severely impacted region is from Africa and Southeast Asia. It mostly infects immunocompromised peoples and actually accounts for more than 15% of HIV-related deaths. Apart from being this successful pathogen, Cryptococcus exhibits two different types of sexual cycle, which makes it the robust model system for us. And the two types of the cycles are asexual, where it grows via budding, and this is how it looks under microscope. And the other part is the sexual cycle, where the two cells of opposite sexes. So the sexes in this fungal pathogen are defined as mating type A and alpha, and I'll refer to them A and alpha in the rest of my talk instead of males and females that we have in humans. So when the conditions are favorable, these yeast cells, they come together, they undergo the fusion, and at this point, the zygote, which is formed after the fusion, undergoes a morphological change and starts forming these filaments. Now, one important part here I would like people, if possible, to remember is that the two nuclei of the parents, they do not fuse with each other until they reach this terminal structure called as basidium. At this basidium, the two parental nuclei fuse, they go through meiosis, and then they form these long spore chains. Now, this is the part of the process that I'm going to talk about in detail. And so I think it is better to give you an idea how this works in the lab. So in lab, we basically mix these two types of cells in a tube, and you spot them on MS media and then incubate and wait for them to undergo the sexual reproduction. The overall time frame, it takes about 2 weeks, where you start with this smooth layer of this one of these spots, and you start seeing these filaments coming out of these areas. And at the same time, after these 2 weeks, if you look closely, you see these spore chains that come out of these basidia. And this is important because these kind of structures I'll keep showing you again and again in the talk. So the process behind this is, like I said, it basically starts when the meiosis takes place. So what exactly is meiosis? For those of you who are not very familiar, meiosis is the stage of cell division where your maternal and paternal chromosomes, the chromosomes that come from your father and mother, they undergo DNA replication followed by segregation to produce 4 haploid gametes. Now, there are 2 fates that these chromosomes can go through. One is there is no crossing over, which basically eventually leads to the same chromosome pattern inheritance in the progeny. But that is usually not the case. What happens in most of the cases is crossing over, where the 2 chromosomes which are homologous from both the parents, they cross with each other, undergo recombination, and then generate the new chromosomes, which have a mix of DNA content from both the parents. And this is essential to generate genetic diversity, basically. Now, this to give you more little bit more idea about how crossing over can affect the germination of these gametes. Now, so if you look at crossing over, you see that the DNA fragments are exchanged. So at the end of the day, you have a chromosome generation where the DNA is sort of intact between both the resulting chromosomes. Now, this is a situation with 1 chromosome, but if you take 2 chromosomes and say if you cross it with opposite sex, which has a translocation generated because of this or because of any other reason, during meiosis, it will end up producing these 4 configurations. Out of these 4, 2 of them will be viable because they will have the entire genetic material in them, but the 2 will not be able to survive because they will be missing either some or the other part of chromosomes in them. So the idea behind this is, if you have 1 chromosome translocation, you expect 50% of your gametes to die if they go through meiosis in this manner. So now this is one thing that I'll again come back at the later point. But so now with this information, let's get into the research part. And to just keep it simpler, I have divided it in 2 parts, where first I'll talk about centromere-mediated chromosome shuffling and speciation, and second is sexual parasitism in cryptococcus neoformans. So for the first part, I collaborated with 2 lab members, Sheng and Marco, who have been very much helpful and instrumental in completing the first part of the story. So now coming to the first part, there are two things that I'd like to explain before the research. One is what is centromere, and what I mean by speciation in this context. And the reason is speciation has different definitions depending on whom you ask. But the speciation term that we will be using today is about the group of individuals which are reproductively isolated from each other, and that's when you start calling them species. Now, this reproductive isolation can be prezygotic and postzygotic. So in case of prezygotic, there are different types, and so if you can have temporal, depending on that they mate at different time periods, ecological, behavioral, and mechanical. So basically they are not able to mate with each other, their gametes are not compatible with each other, and that gives you speciation. The second part is the post-zygotic mechanism, where the two gametes fuse with each other, but then the end product, the zygote, is now either not able to survive or it is infertile, and that's why you basically generate a species barrier in those cases. Now, the next part is about centromeres. So centromeres are the components of a chromosome which are responsible for proper chromosome segregation. It's known as primary constriction site, and in case of mammals, where in chromosomes you can see this one area which is constricted, and that has been defined as centromeres. Now, there are multiple ways of identifying these centromeres, and I am not getting into the detail, but what I want you to understand is that this structure can be highly diverse. And one thing that people have found over the course of time is that the structure of these centromeres are generally comprised of repeat sequences. So whether you take the Schizosaccharomyces pombe, the Physonies, Drosophila, humans, or plants, you in most cases you find these repeat structures, which are can be either DNA transposons or RNA transposons. Now, these are transposable elements which jump from one place to another, and there are plenty of these in most of animals. So when I was a grad student in Bangalore, I identified the centromeric lesions in Cryptococcus neoformans. So there are 14 chromosomes, like I mentioned earlier, and we did some biochemical experiments, and we identified the centromeric locations in each of these 14 chromosomes. When we analyzed these 14 sequences, we found that they were enriched with these 6 RNA transposable elements. So they are named TCN1 to TCN6. There was no pattern of their arrangements. They were randomly scattered throughout the centromeric regions, but they were very specific to these centromere regions. They were not present elsewhere in the genome. Now, when we were doing this, we also identified centromeres in close related species of cryptococcus. So there are other pathogenic and non-pathogenic species, and we found the centromeric locations in all of them. All of them have 14 chromosomes, but when you look at them closely, we noticed that there were a lot of centromere involving chromosome translocations. For example, if you compare B with D, you see these black arrows which specify the centromere-mediated chromosomes translocations, which was very interesting and very curious. And at that point, we thought that maybe these centromere-mediated translocations are one of the ways by which you can eventually generate two different species. So that was the aim of the current study, and the idea was like just break some of these centromeres and see if you can generate a chromosome shuffling phenotype. So to give you a simple schematic, you have these 5 chromosomes to begin with. We used CRISPR and targeted transposons. Then you end up breaking many of these chromosomes in 2 pieces, and then at the end of the day, the cell will fix these breaks which will either give you the original karyotype back, which will match the parent, or they'll give you a modified karyotype where the arms will be exchanged with each other. To finally do this experiment, we targeted one of these elements, which is TCN2, and the reason we chose was because it was probably present in maximum number and was cutting 9 different chromosomes. So we had the tools, we did the experiment, and then we started looking at the transformants more closely. Now this once you get the colonies, the structure, the variation that we are looking at is basically chromosome structure variation. So PCRs and everything else is sort of not applicable when you have to screen this thing. So the approach that we resorted to was pulse field gel electrophoresis. And pulse field gel electrophoresis, you have your cell suspension and your entire the cells, you process them, and then you make the DNA in those agarose plugs, which is then run on these special conditions where the chromosomes are able to separate individually instead of just a small fragments of DNA. So now you look at these, and we identified at least 2 colonies which are named as 136 and 135. And when you look at the chromosomal pattern of these 2, it looks very different from the rest of the transformants as well as from the wild type. So that was something reassuring that changes were happening. Not all the cases chromosomes were going back to their wild type karyotype. That means things were changing. Now the next question was how things are changed and what exactly is the new chromosomal configuration. To understand that, we then did Illumina sequencing and nanopore sequencing. Now Illumina sequencing, it gives you short reads, very accurate, but it doesn't work well when you're dealing with repeat sequences because it everything is very small and repeat sequences are longer. So we went to the nanopore sequencing, where you sequence individual DNA molecules which can be hundreds of kb in length, and this is based on the ion current flow. At this stage, we got the information from both, and we combined it with our pulse field gel information. So based on that, then we ended up with 2 chromosome configurations. So this is the wild type chromosome configuration. We have 14 chromosomes. Each chromosome is represented by a single color. And when you look at the 2 strains that we got, 135 and 136, you start seeing a lot of different variations. So for example, chromosome 1 in 135 is having 1 arm of 5 and 1 arm of parent 2 from the parent. And then the other arms are present in different other places. Similarly, in 136, we had 8-9 different changes. One thing that got highlighted at this point was some of the chromosomes had very weird or not previously observed configuration, and they were very small in size. So we had to make sure that those chromosomes were actually the real entities and not just an artifact of the sequencing. So we ran pulse field gels again at different conditions, and we able to map the band for each of these small chromosomes. Other thing that we noticed is that in both these cases of 135 and 136, the chromosome number was 15. It was number 14. And that would mean there were new telomere sequences that had been added on some of these ends. So we validated those by sequence analysis, and we actually found that there were telomere sequences in the new places which were initially not there, or rather they were actually next to the centromeric regions which are not supposed to be there. And then we looked at the junctions, the new junctions that had generated. So now this is a busy slide, so I'll go a little bit slow. And so what we did was we started comparing the parental centromeres with the new centromeric regions, and we did this in a pairwise manner to actually understand it better. So we started seeing very interesting things at this point. So here you see that a fragment of DNA got inverted, and then you had a new telomere formation. We saw cases where homologous recombination was used to repair some of these pieces. But then we also saw cases where centromeres sequence from one different centromere came and got fused into the junction area, and that happened using non-homologous end joining process, which is a very different type of DNA repair mechanism. So, and then we had cases where the selection marker that we had used was in the centromeres. The Cas9 sequence that we had used was in these areas. So things changed very dramatically at these junctions, and basically the machinery used whatever it could to stitch things together in order to survive. So the interesting part though was that when we looked at these strains and different conditions, they did not show any differences as compared to the wild type. So whether you give them different kind of stress, you put them in a nutrient limiting condition, which is YNB, grow them at high temperature as 37, they are as good as the parental strain, which is H99. We also tested them for their virulence and using this model of Galeria, and we find that the infection basically was as good as the wild type, both H99 and another strain KN99. So they were doing pretty good in mitotically growing cells. They had no severe phenotypic effects on them. The next question was now, what happens when you take them through meiosis? Will they behave, or will they behave as a different species altogether in these cases? Now, the reason for that, like I said, is when you have just one chromosome translocation, you get 50% viability of spores, and the other 50% dies. Now, in this case, we had 8 or 9 different chromosomal changes between the two parents. That would mean that nothing will grow, nothing will happen. So now we did the experiment, as I explained earlier, and we see that and the opposite mating type strain that we have used most of the study is called KN99. This is an isogenic strain to H99, which is the parental alpha strain. So when you cross them, they have the same chromosome configurations, so they mate very well, and you see these nice spore chains. You can also do scanning electron microscope, and you can see that there are basidia which have these spore chains forming onto them. Now for 135, we saw tons of basidia formation, but no sporulation in these cases. And for the second chromosome shuffle strain 136, we saw a mixed phenotype. We had basidia which were not forming any spore chains, but then there were some basidia which were forming spore chains. Now, at this point, we dissected out some of these spore chains, and we saw that 3 out of 96 germinated, which is almost like 3% germination rate. And we were sort of puzzled by this, because with 8 different chromosome translocations, you don't expect anything to grow. That would basically be your species boundaries in that way. So then we started looking at these 3 colonies that grew in more detail. So we went back, did nanopore sequencing for these 3, and basically mapped their chromosome translocations and tried to understand why this was happening. So now you have a scenario where you have crossed this wild type with 136, and you have these 3 progeny. Now, one thing that becomes very clear when you look at these, the chromosome number in these is higher than the wild type, in some cases 16, 17, Other thing that is evident is some of the regions were duplicated. So, for example, if you look at chromosome 12q, it's here and it's also present here. So they were aneuploid and they had the chromosomal configuration which was matching both the parents to a great extent. So it had 11 chromosomes which were exactly as wild type and 11 chromosomes which were exactly as the 136 strain, but then there was a mix of these strains. And as a result of these, when you look at all three of them a little more closely, they all three of them seem to have the same kind of chromosome configuration. So it appears that somehow, in some cases, this particular chromosome configuration is generated, and because this configuration provides entire genetic material to each of the cells, they are able to grow. They are not missing any genes. The only disadvantage is that they are aneuploids. They are not as fit as the parent, but they are able to grow and they are able to manage in those conditions. So with that, I come to my first summary of this, which is basically that we started with a strain which had a wild type chromosome configurations with it CRISPR and broke most of these chromosome arms. And then we had a situation where you have isochromosomes where the two arms of the same chromosome got stitched together. There were de novo telomere formations. Some fragments were lost. Some chromosomes had shorter centromeres. So, and this was enough to give a reproductive barrier with the wild type parent. So, the idea is that just by changing the configuration of these chromosomes, you can actually make a new species. You don't need to change the gene content. In the cases where they were able to mate with the wild type parent, the viable progeny that we got was not perfectly fit. It had aneuploid chromosomes in this, and then it also basically was not growing very well, and obviously it did not mate very well with the parent either. So again, even though some of them were able to survive, they can still give rise to again a reproductive isolation. So the primary objective of our study seemed to be fitting in this scenario. As a but as a side result, what we saw that when we looked at the phenomena that was going on at this stage where multiple things were happening in the cell, we found a very stark similarity of this with a process known as chromothripsis in cancer cells. So in cancer cells, you have, say, this stretch of sequence, it gets shattered, and that can happen because of various reasons in cancer cells. And then you get a rearranged chromosome, which is again completely shuffled. You get chromosomes which are lost, some chromosomes that become circular, and that can happen in both the cases, whether you have one copy or two copies. And we saw a very similar situation happening when we broke centromeric lesions in these cases in yeast species. So one of the future goal for us is basically to see if we can establish this as a system for chromothripsis and hopefully understand that process a much better way using a yeast. And so with that, now I'll move to the second part, which is the sexual parasitism discovery. And this has been a close collaboration with Sheng in the lab. So what exactly is sexual parasitism before I get into the work part? So I'm pretty sure all of you are familiar with the sexual reproduction part, where you have the two parents, they form gametes, and the egg and sperm, the gametes come together, form a progeny, and the process basically repeats. Apart from sexual reproduction, there are some variation that have been well established. One of them is called parthenogenesis and also a form of unisexual reproduction. where the female alone forms eggs, which then germinate and form the adult, which again then form eggs, which again germinate. So it doesn't need a partner. It continues to do this on its own. But then there is this scenario which is called sexual parasitism, and this is called sexual parasitism because in these, one of the parent uses the other partner just for facilitation. It never allows it to pass on its genetic information to the progeny. So there are two well-known types. One is gynogenesis and the other one is hybridogenesis. In gynogenesis, the female forms an egg which has the genetic material from the female, but then the male sperm is required to induce the development of this egg into an adult. The sperm never really forms the part of this progeny, and this is basically what continues and defines gynogenesis. In case of hybridogenesis, on the other hand, you have half of the genome coming from mother and half of the genome coming from father, and they fuse together. They form a hybrid progeny. At this hybrid progeny, when the time comes to pass on the information, one of the genome is excluded depending on the species that you're talking. So in this case, say the paternal genome or the father's genome is thrown out, and only mother's genome is passed on to the next progeny, and this process continues. So the father does, though it is required for the entire development process and it is present everywhere in all the cells of this organism's body, it never really contributes any genetic material to the next round of progeny, and that's why both of them, they are known as sexual parasitism. One thing when you look at this and the occurrence of sexual parasitism is that it is found only in a very few number of organisms. There are few species of insects, fishes, amphibians, and reptiles, and this is where it has been seen so far, mostly in the wild, and we don't really understand it better. So next 10 minutes or so, what I will try to tell you about the story that this kind of phenomena might actually exist in this fungal kingdom as well. So if you recall, I told you that there was one strain known as which we generated using the Wildtype situation, which had 8 changes, 135. And I showed you that where Wildtype forms these nice spore chains, this strain, when crossed with KN99, it doesn't form any spore chains, though it forms nice basidia. This phenomena that we had observed was basically at the time of 2 weeks. So we wanted to see if something changes at a later time point, and around 5 weeks time, we actually started seeing some basidia that were forming these spore chain like structures. The frequency was very low, but they were still there and pretty intact looking. So at this point, I went to Shang and I was like, can we dissect these? And he was like, why not? So he dissected out all these basidia individually, and we started looking at the progeny that was germinating from these basidia. To give you an idea how this looks when you start looking at the progeny in detail. So in case of a wild type cross, which is say H99 versus K99, you have different basidia, and when you dissect the spore chains from these basidia, you get variable germination rate. The progeny is then a mix of both the parents. So some of the progeny will have alpha mating type, some of the progeny will have A mating. And that's what you expect in a normal scenario, because both the parents are contributing to the progeny. But in the cases of 135, we noticed that the progeny that germinated from basidia was only of one type. So for example, the progeny germinating from basidia 1 harbored only mat alpha, and progeny from 3 had only mat A. Now, this was an unusual behavior, an unusual pattern that we hadn't seen before. So we thought of looking at this in more detail. So we started using another mat A parent at this point to do some genetic analysis. And the strain that we used was this known as BT63. So it belongs to a different lineage in the cryptococcus species than H99. So we did the same assay using this A parent at this point, and we found very similar results, that with wild type you have nice spore chains, with 135 you don't have these spore chains, but occasionally you start seeing some spore chains arising. When you dissect the progeny, in case of wild type, you get progeny which has mix of both the parents. But in case of this chromosome shuffle strain, we had only alpha progeny that came from each of these basidia. But what this allowed us now at this point was to understand basically how this genetic information was being passed on. So like I said, BT63 belongs to a different lineage. So it has significant DNA divergence than compared to both H99 and 135. So we sequenced some of the progeny that we obtained here and did whole genome Illumina sequencing. So if you look at the progeny that comes from H99 and a BT63 cross, the progeny has basically DNA fragments coming from both the parents. So if you consider yellow as BT63 and green as H99, each progeny has a mix of both the parental DNAs, suggesting that there is meiotic recombination that is happening between the two parents, and that is how it is being transmitted, which is what you expect in a wild type case. When we did the same analysis for 135 progeny, we see that the entire genetic material is basically coming from only one of the parent. So in this case, 135. There was no contribution whatsoever from the mat A parent in terms of their nuclear genetic material. At this point, we were faced with two questions. One is, does this kind of phenomena happen in a wild type cross? Because we had seen so far only with this strain, which was chromosome shuffle. The other was, if the two parental nuclei are separating as a result of something, then at what stage do they separate? So to answer these two questions, we developed a system, and to give you an idea of that system, so this was what I showed you in the beginning, where the two cells come together and form sporulation. So we thought, if we fill in colors in these two nuclei, we may be able to track it better. So say you put M cherry or red fluorescence color in A parent, and you put green by tagging histone H4 in alpha parent. Now, after they fuse, since they share the same cytoplasm, they will look yellow orangish, and that will continue to grow on and form these spore chains, which will again look yellow orangish. But if something is happening in this process at some stage, what we should also expect is that there will be some basidia which will have only green fluorescence, and some basidia which will have only yellow fluorescence. And that will happen only when one of the nuclei at some point is lost. So we set up this system, and we started looking at the sporulating basidia in both the cases. In case of wild type, as expected, most of the basidia had both colors in them. So you have the spore chains that have nice red fluorescence and green fluorescence. But then there was a very small population that had only one color in them, suggesting that this kind of uniparental sporulation does occur in a wild type population. It was just that we were able to see this in case of 135. And we did the same assay with the chromosome shuffle strain, and as expected, the basidia that had a red and green color in them, they did not have any spore chains. But the basidia which had one nuclei were able to form these unicolor spore chains in them. So, which is basically same as what we had seen with our spore dissection and experiments. So, in order to answer the second question about where does the nuclei separate from each other, we started tracking these kind of basidia which had only one color spore chains and tried to see if we can track down the dynamics of the nuclei. So if you look at this in a wild type case, you will basically see that at all the stages, starting from sporulating till you can go as long as possible, you see these nuclei which have both red and green fluorescence in them. So this is just a scheme, and the last panel which shows that there is orangish color at all stages, and that's what you expect. But in case of the chromosome shuffle strain, when we did this for this particular case, we see that if you track down the red color nuclei, and after this branch point of hyphae, you start seeing both red and green color in the hyphae, suggesting that these hyphal branch points in some way facilitate this biased migration of nuclei and allows that one nuclei to go ahead and form progeny. Now, we still don't know what triggers it, but this seems to be the most convenient way of delivering something into the progeny and then survive in that population. So to summarize this entire story, what I showed you is that you have two parental cells, and in a wild type population you form these biparental spore chains which are yellow in color, but then occasionally there is population which has only one color in this. In case of the chromosome shuffle strain, because this biparental sporulation is no more visible because their chromosomal configurations are so different, you see only those events where one of the parent is going ahead and forming spore chains in these cases. So if you compare the phenomena that's happening in this chromosome shuffle strain with what happens in hybridogenesis, you see some parallel things. So here the two parents come together. Here also the two parents come together, but then at the time of passing on the genetic information, only one of them goes ahead and the second one is left behind. And this is what we see in this cryptococcal neoformance cells also. So our idea at this point is to basically understand this mechanism better and see if we can help and explain the process of hybridogenesis in general in a better way. So with that, I'll summarize my talk, and there are two main take-home messages. One is chromosome structure variation alone can account for speciation, and you don't need to necessarily change genes. You can do that later after your species has been defined. And the second is unusual reproduction seems to be a common trait in fungal pathogens. We already know that cryptococcus also exhibits unisexual reproduction where only the same parent can produce progeny. Then we also know about Candida albicans, which has a very different kind of sexual reproduction case and undergoes what is known as parasexual reproduction. And with what we have identified, seems like there might be another way by which these fungal pathogens thrive and survive in the environment. With that, I'd like to thank my lab, especially Joe, who has been the force behind this entire project and taking it forward, helping out at times, and raising very critical and important questions. And I'll also like to thank the funding from NIH and support from Duke MGM department. And with that, I'll be happy to take any questions. Thank you.
Great. Thank you very much. That's fascinating. It's also amazing to see all this like major DNA rearrangement technology. Let's see. I don't have any questions in the chat right now, but I'll give it a second. We're on a little bit of a lag. So in the meantime, I have a couple questions for you.
Sure.
I guess I'll start with the last part of the talk first. And just like, why do you think that the chromosome shuffled species have more hybridogenesis than the wild types?
Yeah, I don't think it has more hybridogenesis. It's just that we are able to notice it a little better, and the only reason is because everything else is suppressed. So these events stand out in this case. I have done counting of the basidia that form spore chains, like unicolor spore chains in both wild type and in the chromosome shuffle strain, and in both the cases they are pretty low. They are like, if you look at in a cross which has been just 2 weeks old, the frequency is like around 1%, but little less than 1% in both the cases. So I don't think it is more. It's just that these events stand out, and because these are the only ones which are visible to us. Everything else is suppressed. So yeah, I don't think it's more.
Great. Okay, question from Christopher Kennedy. Can you create a MAT A or MAT alpha of the shuffled strain to mate with itself to test if hybridization defects are strictly due to chromosomal incompatibilities?
Yeah, so so there are two main issues with that. One is doing that experiment in a MAT A strain and expecting the same configuration to come out is something which is sort of not feasible at this point, because the arrangements that take place are not in our control, and we can't do that using a normal cross because the mating type chromosome is involved in the one of the translocations. So we don't think at this point that, I mean, it is possible to do it, but it might be a very let's say long process to generate MAT A strain which will have that kind of phenotype. But what we at this point are looking at is a strain which undergoes unisexual reproduction, and we have one strain where you have alpha which mates with itself. Our idea is to basically generate some changes in that and then try to see if, because then it will have incompatibility with the same parental strain, but will have the same exact gene content in it. So but yeah, I don't think at this point we have very efficient tools to generate a strain which will be exactly of the same configuration but will have MAT A in it. So yeah.
All right. Another question from Christopher Kennedy. Lab strains mate and sporulate at high rates under laboratory conditions. Is this trait found in wild isolates of Cryptococcus neoformans as well, or is it a consequence of laboratory domestication?
Yeah, so that was one of the reasons why we did this with another strain, BT63. So we have done this now with 3 different natural isolates. I didn't show the data for that, but it seems like this is a phenomenon that can occur in nature, like in terms of their frequency. But when you take those strains, they are isolated from environment. We have to take them through the lab condition in terms of mating them with the opposite sex partner. But when we do that, we do start seeing phenomena which are similar to these kind of uniparental sporulation events. So I would say it can occur in nature because we have seen that with isolates which are natural isolates and not really made in the laboratory or passaged for a long time in laboratory. So it's a very feasible option for these cells to go through in nature.
Well, I'm going to let that go for a second and see if any other questions come up. I have one more. I was wondering about this, this chromosome breakage from the first part of your talk that you induced. Is there a natural, analog to that process, like something that these organisms would naturally encounter? Because it seems like from your fitness tests, like it wasn't that big of a deal. Obviously, that it made some big changes when you start going through meiosis, but it certainly seems like they've evolved to manage that to some degree. And so I was
Yeah.
wondering, how frequently does that occur in nature? Is that, especially at that level, and, what kind of processes might actually break chromosomes at a centromere?
So I'll tell you that when we started this, we actually wanted to do this with radiation analysis, because radiation is another mechanism which can basically take you through the same process, generate multiple breaks, and then create a lot of chromosome shuffling in it. Apart from the radiation, you have chemicals. There are a lot of chemicals which can break DNA in multiple places at the same time. The only caveat there is that it will not be specific in terms of targeting only centromeres, because when you use radiation, it will basically generate 100 breaks, but it will generate 100 breaks everywhere in the genome. So that's something that I'm pretty sure these organisms do come across when they are in the nature. They go through all that when they are growing up. So it's something that I would say we used CRISPR because we wanted it approach that will specifically target centromeres and not really disrupt any gene functions. But in nature, they may not find that kind of perfect scenario, but there are cases like radiation, chemicals, which can cause multiple breaks. But at the same time, they will have an effect on the genes. So may not be an ideal situation, but definitely if there are obviously chances that a cell will get 10 breaks and all of them in non-essential genes or intergenic genes, that will generate chromosome arrangements. And I think that happens. We know that happens a lot in cancer cells, for example. And for these guys, so we have sequenced a lot of strains now, and we see there are a lot of chromosomal translocations. So only a subset of that is centromere mediated, but there are others which are not really centromere mediated. So
Great. Yeah, that's interesting. I mean, I take your point though. That's probably something that's a little bit more random in nature, but certainly something they encounter. Cool.
yeah.
Well, there's no more questions right now, but definitely check back and see if any more pop up in the comments section.
Sure.
Thank you very much for your contribution to Micro Seminar, and
Yeah.
for all of
Happy
you
to.
out there that are watching. Keep checking our website for updated schedule. We have continual talks coming on down the line. So thank you very much, and we'll see you all next time.
Yeah. Thank you, Cameron.